Okay, so let's start off with the assignment. So it shows autosomal recessive, pedigree chart for cystic fibrosis. What do we know about generation 1? Well, we know that the mom has it and then the dad doesn't. And since it's a recessive disorder, we know that the mom must have both recessive genes. And the fact that none of the kids have it probably tells us that, that none of the kids have it. Probably both of the dominant genes that don't show up, right? So why don't any of the children have it? Well, the dad always gives one of the dominant genes, so that's why it doesn't show up in that next generation. But all the kids will be carriers. So how many boys and girls are in the second generation? Well, we have 1, 2, 3 girls and then 1 boy. This is the mate of this daughter, so only 3 girls and 1 boy. Okay, explain why cystic fibrosis showed up in generation 3. Well, if we build a Punnett square like this, all of the kids are going to get one from their mom and one from their dad. So obviously this person has a heterozygous as well, and that gives them a 1 in 4 chance of having a kid, and they got that 1 in 4 chance. Okay, it's a pretty easy one to analyze there. So this is albino, autosomal recessive, another autosomal recessive. So what are the genotypes for this first generation? Right, so the dad has it. And then some of the kids have it, which probably mean that the mom is a carrier for it. Genotypes of the offspring, if we do a pedigree chart. Right, so little r down, big R, and across like that. So the kids that have it are like this, and then the kids that don't have it are carriers. Okay, any more questions? Nope. So what do you notice about all the offspring that are affected? They're all males. Explain why this is happening. Well, this is probably going to be sex-linked, and the boys only have 1 X chromosome. The fact that their mom had it makes it even more likely because their mom gives them that X chromosome, and then the dad gives the Y, so there's no backup. So it's a 100% chance that all the boys are going to have it if a mom has it, and it's sex-linked. Okay, so define mutation. Why does this start over? Okay. It's interesting. I didn't realize that it started over. Sorry. Define mutation. So this is a typo in DNA. Explain how people who are carriers of the allele for cystic fibrosis do not have symptoms while they have 1 good gene and 1 bad gene. So this one is dominant because cystic fibrosis is a recessive disease. It doesn't show up if you have the dominant, the good allele. Okay, and this shows red-green color blindness is a sex-linked trait. The pedigree so it's sex-linked, so that's important. Okay. In the first generation, is the father or mother colorblind? Determine the number of males and females produced by the father and mother in the first generation. So we have 1, 2, 3 males, and 1, 2, 3 females. So you can see they showed us the carriers, which makes it a lot easier. They don't. I won't do that on the test, but it's just to analyze it. Save the number of individuals in this pedigree who are carriers for color blindness. 1, 2, 3, 4, 5, 6, 7. And how many males and how many females have color blindness in the pedigree? So we have 1, 2, 3, 4, 5, 6. I guess 1, 2, 3, 4, 5, 6. 6 males have the color blindness, and then only 1 female. And you can see that in the female that got it, the mom was a carrier and the dad had it. That's the only way for a female to get it, is the dad has to have it and the mom has to be at least a carrier for it. So this talks about sickle cell anemia. It's an autosomal recessive disorder. Very important information. So the impaired genes causes red blood cells to be shrunken and sickle-shaped rather than normal disc shapes. These deformed red blood cells can block narrow blood vessels. People with two recessive sickle cell alleles become very ill and die at a young age. Most people who suffer from sickle cell anemia or carry the recessive gene tend to form in areas affected by malaria, which is a deadly disease carried by mosquitoes. The malaria parasite reproduces. People who are carriers of one of the mutated alleles actually have an advantage over non-carriers. They are resistant to malaria. So this is important. So it looks like they're using S's. So this means that you are a carrier, and this means that you cannot know malaria, right? Because it's recessive, this means you have it. You have sickle cell. And this means no malaria. And then if you have two big ones, that means no sickle and yes malaria. Okay, so this is really interesting, right? Because these people here will probably die of malaria. These people here might die of sickle cell, right? These people here, they don't get malaria and they won't die of sickle cell, but they pass it on to their children, so that's bad. If they pass both of the genes on, but this is like the perfect possible scenario for people who live in areas affected by malaria. So use a Punnett square to show the results of two heterozygous people. So two people, these are the people that are likely to survive because they won't get malaria and won't get sickle cell. So little s down, big S down, little s across, big S across. Okay. What are the genotypes of the child? Well, all genotypes are possible. State the probability that the child will be resistant to malaria. 75% chance of being resistant to malaria because this is the only one that can get malaria. State the probability the child will be developing sickle cell anemia. There's a 25% chance. And explain why sickle cell anemia allele is more common in areas affected by malaria. That's because anyone like this will probably die of malaria, right? So these people die of malaria. They don't reproduce. These people are more likely to reproduce. So that's why there's more people with sickle cell anemia. Is the following autosomal or sex-linked? Is it dominant or recessive? So none of the parents have it, and the kids have it. This is twins, by the way, which means that it's not dominant. It's going to be recessive. In terms of sex-linked, both boys have it, and then the girl doesn't have it. Probably sex-linked. That's the best information. It doesn't give us a lot, so that's not really helpful. If we had another generation, we could tell a lot more information, but for what we can see, there's definitely a high possibility that it's sex-linked. Is it following? Yeah, so same thing. Okay, so mom has it, boys and girls have it, so let's get out sex-linked. In terms of dominant or recessive, well, let's take a look. So let's look at recessive first. So if it's recessive, if it's recessive, then that's what this one has. And the fact that some of the kids get it mean that this is probably heterozygous, right? This kid gets it, this kid does not get it. Or this kid gets it, this kid does not get it, so there's a 50-50 shot. So yeah, definitely like that. If we build a Punnett square for this, these are the two possible genotypes of the kids. So this one has it. This one's a carrier. This person has it. And then they have one kid with it, one kid without it. Yeah. So definitely could be recessive. I don't think there's anything, if we go a little bit further, yeah, some of the kids get it, some of the kids don't get it. In terms of dominant, if it was dominant and we took a look at this, let's erase all this. So dominant, if this person has it, so this person doesn't have it, which means that's what they are. And then this person could be this, but because some of the kids don't get it, they have to be like that. So same sort of path, it'd be the same Punnett square. I don't think there's enough information here to tell if it's dominant or recessive. It could be either one. It would have to like skip a generation or something like that. So definitely not sex-linked, could be dominant or recessive. And then this last one, parents don't have it, right? Which probably means, oh, but all boys are getting it. So all boys are getting it, which means it's probably sex-linked. Yeah, if all the boys are getting it. Okay, so that's the homework. So let's move on to the sample diploma question. So based on the pedigree chart, the pattern of inheritance, kind of like that last one we did, both parents don't have it. Boys and girls get it. It can't be dominant with both parents not having it, right? It has to be recessive. That's a big giveaway for that. And the fact that boys and girls get it means that this is out. So it's probably recessive. Both parents are probably heterozygous, which means there's a 1 in 4 chance of their kid getting it. And yeah, that's what we see. None of their kids have it, but this one has it. And then this person has it, which probably means that, so this person has it. This one's a carrier for it, which means that that's why that kid ends up getting it. And then this kid's probably a carrier, right? If we do a pedigree chart, let's just do a pedigree chart, or sorry, a Punnett square. Right? So there's a 50% chance that their kids are carriers, and I think both of their kids are carriers. There's only 25% chance that their kid gets it, their kid doesn't get it. And then there's a 25% chance that their kid is not a carrier for it. So yeah, I would say autosomal recessive would probably be the best answer for that. Okay, and then this question, which of the following pedigree charts best represents the inheritance of Huntington? So Huntington is an autosomal dominant disorder. Okay, neither the man's nor the woman's mother have Huntington disorder. So man's, so this is a man. So neither the man's nor the woman's, or it's talking about, sorry, this man and woman. So this man and woman, and this man and woman. Okay, so neither the man's nor the woman's mothers have it. Okay, so this one's works with this. Mothers have it in this one. This one works for that one. And then one of the mothers has it. So we're down to a 50-50 shot. And let's go to the next one. The woman's father and brother have Huntington's. So the woman's father and brother. Good. The woman's father, this one doesn't have any brother. So it looks like A is the answer from there. Let's just check the other ones. The man's 2 sisters do not have Huntington's. Yeah, that's good. And the man's father has Huntington's. Yes, A is the correct answer. Resistance to antibiotic drugs in bacteria populations have been observed over time. One of the reasons that bacterial populations develop resistance to antibiotics is, so we talked about that, the two ways that they can transfer DNA really quickly is through plasmid transfer and transformation. So those are the type of answers we're looking for. So that's C. Okay. So we're looking for a responding variable for this. So the thing that changes because we change something. A researcher is studying development of antibiotic resistance. The researcher sets up 4 different experimental groups. From the 4 groups, they're exposed to antibiotics as described below. The 4th petri dish is a controlled group. So doses of increasing strength of antibiotic on E. coli, doses of different antibiotics on E. coli, and doses of antibiotic that kills the E. coli are applied a number of times. So it looks like they want to see what's going to kill this E. coli, right? Does this kill it? Does this kill it? Does this kill it? So the responding variable is the surviving E. coli. Does it kill it or does it not kill it? Okay, based on the pedigree chart, the mode of inheritance is, so not sex-linked, because boys and girls get it. Right here is the key to this one. So if it was recessive, this would be the genotype, right? Which means that all the kids would have to have it. But if it was dominant, right, they could be like this and they could have it. So that means that if we do a pedigree chart with that, right, they have a 25% chance of the kid not having it. So they have 2 kids that not have it, and a 75% chance of the kids having it. So yeah, it's got to be dominant because of that right there. Yeah, if it's recessive, if both parents have it, all the kids have to have it. Okay, so let's get started in this lesson, genetic technologies. So describe in general terms genetic engineering and its application to gene therapy and development of genetically modified organisms, and explain the decisions regarding the application of scientific and technological development in perspective. So you need to know these perspectives. Okay. So we'll talk about these perspectives. So one, a viewpoint, you can have a social and cultural. How does it affect the group of people as a whole? How does it affect our traditions? Environmental is how does it impact the environment? That one's nice and easy. Ethical is it good or bad to do? And economic, does it make us money or not? Okay, so I'll give an example for all of this. So my example is we have thanks, let's switch those, giving. And during Thanksgiving, Canada decides to ban turkeys. No turkeys, you're not allowed to eat turkey. You have to go vegetarian on Thanksgiving, okay? So we can talk about it from all these different viewpoints. So let's talk about it in terms of ethical. That's a very common one. So is it okay to kill animals and eat them? Are farming practices okay to do? Is it unethical to sort of put them up in these coops? So is it good or bad to do? Ethical, it's probably a good decision, right? It's to not kill those animals. It's lots of people would say ethically this is a good thing to do. Okay. In terms of economic, this is bad, right? A lot of farmers rely on selling turkeys at Thanksgiving to make money. So this is not good in terms of economics because you put people out of jobs. So that would be bad. Social and cultural, right? Thanksgiving, is the turkey, right? How does this affect our traditions, right? So our traditions are to have Thanksgiving. People aren't going to be happy about that. So this is bad for our traditions. A lot of people would be upset over that. And environmental, how does this impact the environment? Well, raising animals is actually pretty bad for the environment. We put a lot of energy. Greenhouse gases are created. Yeah, so this is probably good for the environment. So it's good ethically, good for the environment, but bad for those. And you're never going to get something that comes up that is good from all viewpoints. And it's really important that you think of things from viewpoints other than your own, because what you think is not always correct. Okay, so those are the viewpoints. So let's talk about different technologies. So DNA fingerprinting works on the idea that every cell in the human body has 23 pairs of chromosomes. So if you find a cell at a crime scene, blood, saliva, skin, hair, you can map that person's DNA from it. So when they collect that DNA, they put it in a gel, they swirl it, they use an electric field, and an image appears, and this is what the image looks like. So what they would do is they would take the victim to sort of eliminate that DNA, take different suspects, and this is evidence from the crime scene right here, and match it up with, well, what suspect matches up well? Well, suspect 2 has the exact same DNA fingerprint, so suspect 2 probably did it. Another use of this in non-crime scene is DNA art. So I'll open up. This is a web page that you can order DNA art. So you can choose your different one. You can do fingerprint. So maybe you want 2 people. You want a horizontal one. You want a big one. Oh, it's expensive. Let's add the nicest frame on that and colors. So it's very, very expensive to do this DNA art, but it's kind of cool. Have your DNA up on the wall. Who knows if it's actually your DNA? So here's some questions I want you to think about. Should DNA for every person be collected in order to increase national security? So we have to think about those different viewpoints, right? Is it ethical? That's probably the biggest one for this, right? Is it ethical for the government to have your DNA? Would it help solve crimes? For sure. But don't you have a right to some privacy? And who's getting this DNA? What are they doing with this DNA, right? Are they doing tests on it? Who knows if the person who has access to this is ethical, right? So that's what makes it difficult. Should parents test the DNA of an unborn child using amniocentesis, right? So again, this is ethical views with this. It's not really environmental or maybe traditional with religion and things like that. But usually if they find something that they don't like from the test, they go to terminate the child. And is it okay to terminate an unborn child or not? That's, everyone has an own ethical decision. When does life start? Is it okay to end a life? That's definitely a tricky one. And should a person be allowed to refuse a DNA sample to authorities? Again, are you allowed to have your privacy? What if it'll help solve the crime? What if the person did it and they won't, and they aren't allowed, or they're allowing them not to test their DNA? Lots of things to think about. So transgenics is the process of intentionally altering the DNA of an organism by transferring DNA of one species to another. So here in this picture are some mice, and you can see 3 of them are glowing because they have been given DNA from a jellyfish, and that allows them to glow. So genetic engineering is a modification of genetic material through actions of selective breeding or transgenics. And a GMO is an organism that has its genetic material deliberately altered through transgenics. Okay. So this is genetic, engineering. Oh man, that's so bad. So this is transgenic engineering. There's 2 different ways that we can do it. We can do transgenics, and then we can do selective breeding. Right, if we do selective breeding, we get dog breeds, the different dog breeds. And if we do transgenics, that's when we get a GMO. Okay, so here's a couple videos that show the positives and the negatives of genetic engineering. One of them is bad because it's like, yeah, it gives some false information and makes you, yeah, what's the word I'm looking for? It's propaganda that isn't always correct, and they don't do it in a very scientific way. And the other one's pretty good, but they don't talk about any negative downsides. So watch these two videos, and hopefully they work and don't get cut out on YouTube because of copyright issues.
So you want to know how a GMO is made? You're not alone. It's one of the top questions about biotechnology. There are different ways plants can be genetically engineered. We'll use the story of the papaya in Hawaii to walk you through one of them. Step 1. Determine whether genetic engineering is the most effective way to solve a plant's problem, such as a disease or damage from pests. For centuries, traditional plant breeding has been used to fight pests, environmental stress, such as drought, as well as to improve taste and nutrition. The reasons to develop GMOs are no different today, but GMOs can be created more quickly and more precisely. Other breeding methods can take decades to produce results and aren't able to be as precise in focusing on one genetic trait or isolating a solution to a specific problem. Since the 1940s, the Hawaiian papaya industry had been battling a problem, the destructive and rapidly spreading papaya ring spot virus, aka PRSV. PRSV deforms the papaya fruit and in young plants destroys the plant's ability to produce fruit at all. PRSV is transmitted by insects that feed on the fruit. Many methods were deployed to try to stop the destructive virus. In the early 1960s, farmers moved papaya production from one Hawaiian island to another, from the island of Oahu to the Big Island of Hawaii in the Puna district. The virus ultimately reached Puna. Other methods didn't work either, including using insecticides for the insects spreading the virus, giving the plants a mild strain of the virus to create natural inoculation, or destroying infected trees. The virus just continued to spread. 53 million pounds of fresh papaya were being produced annually when PRSV was first discovered in Puna in 1992. By 1998, that amount had been cut in half to 26 million pounds. The crops and livelihood of farmers were being destroyed in front of their eyes. Moreover, for consumers, the availability of fresh papaya and papaya juice for fruit, salads, smoothies, and other products were threatened. A powerful new tool, biotechnology, was now available to fight back. Farmers and researchers turned to genetic engineering, beginning in 1985, for help. Step 2. Identify the gene or genetic material that could solve the problem, like providing resistance to the disease or pest, and study the genetic makeup of the plant that needs the trait. Researchers and farmers had to find a way to make the papaya plant resistant to the deadly virus. PRSV resistance was the genetic trait needed to solve the problem. Researchers knew that if plants contained certain genetic material from the virus within their own DNA, they were protected. If they could insert a copy of this gene from the virus itself into the papaya's genetic makeup, the plants would be protected. Step 3. Copy the trait from a donor organism and implant it into the plant's DNA. Researchers isolated a PRSV gene from the virus itself. Then they needed a way to get the genetic material into the papaya plant's own DNA. Researchers deployed a commonly used technique in biotechnology called transformation, a process by which a copy of genetic material from a specific trait is inserted into cells. There are many ways to transform a cell. In this case, the researchers used a high-speed particle bombardment method. Through a high-pressure insertion device, the gene from the virus was inserted into the DNA of the papaya seed. The genetic material made the plant resistant to the ring spot virus, just as a flu shot makes people resistant to the flu. The researchers were able to successfully transform 17 plants. These plants contained this gene from the virus and produced a protein from the virus that helped the plants become resistant to the PRSV infection. Step 4. Plant the new seed and test it. The final step was to plant the seeds and carefully test the resulting plants. One line of plants demonstrated consistent resistance to the virus. After several years of testing, it was determined that this line performed well on the farms, was safe for people to eat, and was of commercial quality. Success was achieved. One variety of transgenic papaya, the rainbow, yielded about 125,000 pounds of marketable fruit per acre per year, 25 times the amount of fruit produced per acre by a non-GM variety infected by PRSV. These tests took about 7 years, including food safety and environmental review by the FDA, USDA, and EPA. Once regulatory approval was gained, the Hawaiian farmers chose to start using the new papaya seeds. The result? The harvesting of the new PRSV-resistant papaya began in 1998, and by 2001, the industry had rebounded, producing 46 million pounds of fresh papaya, free of PRSV. This kept the vibrant papaya industry alive in Hawaii and ensured consumers had access to high-quality papaya and papaya juice at a reasonable cost. Genetic engineering allowed the introduction of PRSV resistance without altering other traits of the papaya or changing the plants in other ways. The plants were exactly the same in nutrition and safety. As of 2010, 70% of Hawaii's papaya industry is GM papaya. GM papaya has been grown and consumed in the U.S. for more than a decade. This is just one GMO story. We hope to share more with you. Please share your comments.
Genetically engineered crops in agriculture, a worldwide experiment on people, animals, and nature. Cultivation of genetically engineered crops like soy, maize, cotton, or canola have far-reaching consequences. 90% of genetically engineered crops belong to the corporate agricultural group Monsanto. The rest are owned by Syngenta, DuPont, Bayer, and others. Genetic manipulation of crops can have uncontrollable consequences. The function of genomes is only partially understood, but foreign genes are still introduced into crops. These genetically engineered crops are widely cultivated outdoors, where they cannot be controlled. Currently, 90% of GE cultivation occurs in the Americas, particularly in the USA, Argentina, Brazil, and Canada. It seems researchers have forgotten that genetically engineered crops once outside can no longer be controlled like they could in the lab. GE crops can self-replicate and pass on their new characteristics to neighboring crops, penetrating the fields of farmers who want to cultivate their crops without genetic engineering. Adverts from the genetic engineering lobby claim genetically engineered crops produce higher yields, but this marketing mantra is a complete hoax. It has been shown that crop yields for GE crops are no higher than normal crops, but farmers must buy more expensive patented genetically engineered seeds each year. This forces them to become dependent on corporate giants. But that's not all. Genetic engineering giants also produce pesticides and herbicides. There are two characteristic traits of genetically engineered crops. They are either resistant to herbicides, in which case the genetic engineering company itself produces the suitable herbicide, or the genetically engineered crop emits an insecticide. Whether or not the crops are resistant to weed killer or give off poisonous gases, not only weeds or pests are eliminated, other beneficial field growth and some living creatures are killed as well. But nature adapts, so pests and weeds develop resistance to pesticides. And that means that increasing amounts of stronger pesticides will be used. Especially grave consequences are observed in this respect in monocultures. For example, in South America, rainforests and other natural landscapes are destroyed every day to make room for genetically engineered monocultures and pasture. At the same time, the soil and groundwater are poisoned. On top of that, the genetic engineering lobby claims genetically engineered crops can help to reduce world hunger. Genetic engineering is not the answer. There is enough food on Earth. The problem is that it's not distributed fairly. But the fact is that most genetically engineered crops find their way into the livestock chain, so they end up as meat or dairy products in supermarkets. Along with all the destructive effects on the environment, one last awkward question remains. How do genetically engineered crops affect humans and animals? The answer is, no one knows. Therefore, it's important for you to carefully consider which products you buy. Get informed and get involved, because our world is not a testing ground. For more information about genetic engineering, go to greenpeace.org/GMO.
So it's important to, take both videos with a grain of salt. The one that says it's bad is, it shows like rat poison and biohazardous symbols, right? It has some valid points with crops spreading to other crops, genetically modified crops spreading to other crops, and then people owning patents. That's not good, but it does it in a very unscientific way. And there's lots of bad information on the internet. It's really important that you don't just watch one video and then set your viewpoint and never change. It's important to look at all different perspectives and look up more than one piece of information on something. So these are benefits of GMOs. GMOs are good, right? Crops can produce their own pesticides. That's awesome. That means you spray it with less pesticides and it's good. Another one is bacteria containing a human gene of producing insulin can be given to diabetics. So instead of bacteria being bacteria, you make it produce insulin, and then people don't have to get shots every day, and you basically cure diabetes. That can be done through transgenics. Pig with human genes, that produces antigens to make pig organs compatible with humans. So you can put human antigens on pigs. So when you need a kidney, you can just get a pig kidney and your body won't reject it because it'll have the right antigens. That would be incredible. This one's kind of weird. Goats can produce milk with spider silk in it, making very strong ropes. So you make your goat milk rope. And this one's kind of cool. An evergreen tree with jellyfish gene can make it glow. How nice would that look at Christmas? Obviously, you wouldn't want that released into the wild because you have these glowing forests, screws up ecosystems with nocturnal animals. That one would definitely have environmental downsides to it. Okay, biggest downside of GMOs is unknown effects to the ecosystem. So they've made herbicide-tolerant canola. It can pollinate with weeds, and it makes them herbicide-tolerant, so that's not good. Then what do you use to kill the weeds? And corn, that produces its own pesticides and kills the monarch butterfly, which is bad for the ecosystem because monarch butterflies are responsible for pollinating things. Bioweapons could be produced using a disease combining traits of 2 or 3 pathogens. So that would be really bad if some unethical people got their hands on this technology. Okay, this is something else you need to know, gene therapy. So diabetics cannot produce insulin, so they take injections of insulin multiple times a day. So gene therapy is using a vector, such as a virus. So a vector gets a disease into something. So they use a virus to repair or replace genes that can possibly cure genetic disorders. So if you remember when we talked about viruses, what viruses do is they take a cell of a person and they come in there, they inject their DNA, and their DNA produces more viruses, right? The DNA tells the cell to start producing more viruses. But what you could do is instead of that information telling the cell to produce more viruses, that information could tell the cell to, hey, make insulin, repair genes, right? And you inject enough of these viruses over time into all the cells, they change the DNA of all the cells, and the person with a genetic disorder doesn't have that genetic disorder anymore. You rewrite their DNA. So scientists take away the disease-causing agent of the virus and use its ability to take over cells and reproduce using them. And over time, the virus would replace a faulty gene with a working one, and you cure someone. Okay, so here's a video that talks about gene therapy.
Gene therapy has been studied as a potential treatment for genetic disease since the 1980s. The goal of gene therapy is to help correct the genetic disease by providing a functioning copy of the gene to make up for the genetic defect. In the laboratory, a functioning copy of the gene is packaged into a lentiviral vector. This is a modified virus that acts as a transport vehicle for the functioning gene. The lentiviral vector delivers the functioning copy of the gene into stem cells taken from the patient. The gene is then inserted into the DNA of these cells. In recent years, scientists have made improvements in these vectors and continue to study gene therapy as a treatment for many different diseases, including cancers, blood diseases, central nervous system disorders, and immune system diseases. More than 1,800 clinical trials have been conducted using different types of gene therapy in various diseases.
Okay, so we're going to do these sample diploma questions now, so pause it and try them out on your own. An experimental treatment for PKU involves injections of a virus that contains normally functioning gene. That is the definition of gene therapy. Okay, a type of transgenic mouse called the Harvard mouse is used in cancer research. Harvard mice are born with a gene from another organism that makes them susceptible to develop cancer. So that would be genetic engineering. They altered the DNA of the mice to make them get cancer so they can test things on it, right? This one here, the statement that best supports the production of genetically modified salmon. So this is important because lots of people get the right answer when we're talking about environmental, but remember that it's saying support. So lots of people pick genetically modified salmon could escape from the wild, disrupting. Yes, that's an environmental one. Farming genetically modified could increase food supplies, not environmental. Some people believe that genetically modified salmon triggered allergies, not environmental, and farming genetically modified salmon could reduce overfishing. That is environmental. So 2 and 4 are both environmental, but 2 is saying it's bad, so it doesn't support it. 4 is saying, yes, I do support it, and that's why D is the better answer. Okay, and then let's go through the assignment for this. Okay, so match up these terms. I'll call this 1, 2, 3, 4. A farmer uses a plastic bag to collect pollen and the fastest growing corn plants. So that looks like selective breeding. Enzymes are used to cut up DNA left at a crime scene, so that's DNA fingerprinting. A modified virus is used to deliver a defective gene, that's gene therapy. And a gene in a bacteria that produces a protein with insecticidal properties, so that is transgenics or genetic engineering. List some potential advantages of genetically modified crops. They produce their own pesticide, right? They can have higher yields. Maybe they use less water. They're drought resistant. One risk of applying transgenics to a new type of organism, biggest one with that is you don't know how it's going to affect ecosystems. Also, people eating it. Can we eat it? Is it safe for us to eat? Explain why virus vectors are used in gene therapy. So virus vectors are needed because they can inject DNA into the cell and take over the cell's reproductive machinery. So it produces what the virus wants. That's why it's important. With this one, samples from crime scene. Who did it? Well, just looking right here, it looks like suspect 2 did it. And then 6, most citizens from Iceland have volunteered their genetic information. 1 risk and benefit with a nation possessing a gene bank. Risk, is it going to be used for good? Is it going to be private? Will they sell it to someone, right? And then a benefit for it is if there's a crime, they can solve it really easily. If there's genetic disorders, they have more information to sort of find those genetic disorders, things like that. Okay, that is it. And if you have any questions, let me know.